you know him through many different ways, but one of the ways is he's editor-in-chief of the flagship journal in our field, Asia Synthetic Biology. So if you have any questions, especially students, how to publish papers in Asia Synthetic Biology, just approach him and ask him how to do it. So today his talk will be shown here. It's gone. It was pushing the scale of something, right?
Yes.
Okay, Chris.
Great, thank you, Sangha. There we go. Great, well, it's an honor to be here. I was lucky enough to come to the last synthetic biology meeting in Suzhou a couple of years ago. And the way I wanted to start this talk is really describe, I think, some of the ambition of synthetic biology and where I think things need to go from where we are today to really thinking out 10 or 15 years from now, what types of things have to be put into place. And how we have to get much grander, I think, in our vision of how we can use biology to build ever more sophisticated things. And so, if you really look at what the field is doing now, I'm showing a number of different products that some of the real bleeding edge companies in synthetic biology are. Does that help? I'm trying to see if this is on, maybe. So some of the products that are coming out, and these are really things that are not even yet on market. And so these would be big successes if they were to actually come out. So things like isobutanol, which companies like Gevo and DuPont are working on. Genomatica has a BDO process. Amyris has been doing pharmacine. And then a company out of my lab has been making spider silk proteins as part of spinning them into fibers. So if you look at all of these different products, the thing that's shared amongst all of them is that they're actually very simple chemicals. So there are a few carbons, they're individual proteins if it's a protein-based products. They're really very simple. And really, from the perspective of drop-in type replacements, the things that we already derive out of petroleum-based products. And so if you take a moment and just contrast the types of things that we're currently getting out of biotechnology and synthetic biology with what some of the potential is of being able to engineer biology, there's a big discrepancy. So in the area of materials, if we look at what types of materials you can get out of the natural world, there's this incredible sophistication and complexity that's possible. So I'm showing some examples of that, and these are natural things that biology is already able to make. So if you look in the upper left, that's a diatom that's able to build this very precise nanometer and micron scale structure that has a level of precision that we just don't have the capability to be able to control when we're engineering biology. And there are all different types of shapes and structures that are possible. There are sponges that live at the bottom of the ocean that are able to essentially spin fiber optic cables out of silicon. And really the types of things that biology is exceptionally good at are being able to take materials and be able to derive them from the nanoscale features all the way up to macroscopic properties. So if you look at a tree trunk, a lot of the very fine detail, you have both the detailed scale of the cellulose fibers of the trunk itself, but then you're able to get a macroscopic property out of that. And that's something that we just don't have the capability of being able to control when we're engineering biology. So that's materials. So if you look at chemicals, if you're going to go beyond the isobutanols and the very simple carbon-based chains, they're extremely sophisticated chemicals that can come out of biology, and I'm just showing two of the many examples. So madotoxin is a natural product that's made by an organism that lives in the ocean, and it's a very potent toxin. And immediately when you look at this and compare it to butanol, this has an incredibly sophisticated structure, has very careful precision. It would be very difficult to produce something like this using tools from organic chemistry or to derive it out of petroleum. There are also molecules that are very difficult to produce chemically, like lateranes, which is made by an organism that actually has to deal with hydrazide. And this is a potential rocket fuel. And so if you look at these types of molecules, again, it's something that's very difficult right now to be able to engineer and not just utilize something that's naturally being produced by biology, but to go in and try to engineer from scratch cells that can build chemicals with this type of sophistication. And then finally, I think one of the ambitions of the field is to try to be able to program cells as part of being the product themselves. And so this is a vision that we had a number of years ago in trying to be able to engineer cells that would act as therapeutic agents, where you would take a bacterium and take its, this could be annoying, its natural, one second, let me try that. And try to utilize its natural ability as a pathogen to be able to identify different microenvironments of the human body, but be able to harness that, where you would take a bacterium, program it to have the ability to be injected as a therapeutic, identify a place where a tumor might be situated or some type of therapeutic intervention is needed. It's able to swim around in the body, identify the right environment, and then be able to deliver a therapeutic response at that environment. And so in order to do this, it's not just enough to try to engineer the cell to be able to make a product or make a chemical, but you're really trying to treat it as a sort of micro-robot where it's able to receive information from a number of sensors in the cell, process that information using circuitry, and then implement a number of different responses. So what I showed is that currently in the field, we're talking about very simple types of products where it's just making a small molecule, and that's very difficult as it is, but we really want to span to these much more sophisticated types of things that we could engineer biology to make. So from the material structures of diatoms to the incredible natural products that we see in the natural world that could be used as therapeutics, to even using cells as programmable therapeutics themselves. And so you can look and say, well, what is needed by the field now in order to go from these very simple products to the much more sophisticated things that we're envisioning? So one thing that we need are new design tools. So when we think about a genetic engineering project, we still think very much at the scale of taking units of DNA in a program such as Vector NTI or some sort of vector building software suite and physically putting together these pieces of DNA. And that really causes us to have to build very small systems that don't have a lot of pieces that we're trying together or very sophisticated functions. And we need completely new paradigms for thinking about how to create much larger projects in genetic engineering and how we can utilize computers to help us both with some of the decision making and to identify problems before they happen. We also need synthetic regulation. So gene circuits remain one of the most difficult things in synthetic biology in order to make robust. And we need them because in these much more sophisticated programs, we have to be able to control when and where genes are turned on as part of building a big material or creating a complex chemical or being able to identify what region of the body you're in and have an appropriate response. And then finally, all of these different systems are common where you need to be able to manipulate many genes. So currently in genetic engineering, we're stuck with 6 or 8 gene systems that we can introduce into a cell. And we really need to be able to push that, where there are hundreds and thousands of genes as part of genetic design. And we have all of the parts to be able to control those genes. We have the regulation to tell them when they turn on, how we can insulate such a large system from the natural host, and then ultimately be able to figure out what happens when something goes wrong and really at the scale of the cell itself be able to debug problems. So what I'm going to do today is go through how my lab is trying to address some of these different issues and how we're trying to really think from the top down how we could go about taking on much larger engineering projects and what are some of the foundational types of tools that we need to be able to push the scale at which we think of the process of genetic design. And so this is what I refer to as genome-scale design. And I'm not exactly referring to being able to build an entire genome from scratch, but rather, if we were to try to take on genetic engineering projects where we put in a megabase of synthetic DNA, so something that's really on the scale of the genome, what are the types of things that we have to account for? How do we even think about organizing or modularizing a design of that sophistication so that we can approach the different aspects independently? And so at a very high level, this is the paradigm that we're thinking of, where we currently will split a design into 3 distinct parts, and that allows us to be able to think about what the regulation is that's necessary, how we have multiple multi-gene systems that have to be controlled, and then how we can link the whole system together. And so the 3 different areas, and I'll talk about each one today. The first is controlling when the different genes are turned on, and we call this the controller. And so this is a distinct region of the genetic design that contains the synthetic sensors and circuitry to tell the cell when the different systems should be turned on as part of this larger program. We then have the actual, what we would call actuators. And so these are the different things that the cell can do. So for example, in this hypothetical example that I'm showing in this slide, this is envisioning a bacterium that can spin an antibiotic cloth. So a silk fiber that has natural product antibiotic embedded within it. And so that's something that's not that sophisticated, but much greater than what we could do now. And so you might imagine, well, we have to have large silk genes, and so that's one actuator. Those silk genes have to be pumped out of the cell at some time point, so we need a protein secretion device. The antibiotic itself is a complex natural product that has multiple genes involved, and then maybe when you're done, you want the cells to kill themselves. And so these are all different actuators that need to be turned on at different times, and so they need to be linked to the controller that has the regulation for defining when those times are. And then one of the newer ideas that we've been having is what we call a resource allocator, and that links the controller to the actuator and provides for all of the different components that the synthetic system needs. So it says how many polymerases are available to it, how many ribosomes, and so on and so forth, and is a mechanism for linking the circuitry. So first I'm going to talk about the controller and some of the things that we've been doing to advance it so that we can design regulatory systems that ultimately we want to be on the scale of the natural regulatory programs in a bacterium. So first, for sensors, our lab has developed a number of different genetic sensors where we define a sensor as being a unit of DNA that has all of the necessary components where when you put that unit of DNA into a cell, it gives that cell the ability to sense something and turn it into a transcriptional response. So for example, if you have an oxygen sensor, then there would be a unit of DNA that has all of the necessary proteins to sense oxygen, and then the output of that sensor is the activation of a promoter. And so for each one of the sensors, whether it's an acetate sensor or a sugar sensor, in all cases, they're turning on a promoter as you add that stimulus to the cell. And that's a really important point because when you design the sensor so that it activates a promoter, that then becomes very easy to connect into genetic circuits that allow you to process the information that's coming from that sensor. And we've been working on very simple modular circuitry that allow us to take the information of the sensor and then be able to make different decisions within the cell. In order to do that, we designed a very simple computational element, a NOR gate, which allows us to both receive information that's coming from a sensor in the form of an activation of a promoter, and then allows us, by connecting NOR gates in different configurations, to make any type of signal processing program that we would want in order to integrate those sensors. And so a NOR gate is very simple. The NOR gate is on when both the input signals are off, and then is off otherwise. And so you can make this very simply in a genetic system by having 2 promoters that serve as inputs. And these could be promoters that are from the sensors, or they could be promoters from other circuits feeding into it. They then turn on a repressor of some sort that then turns off a third promoter, and that third promoter is the output of the system. And there are a lot of different biochemistries that you can use to have this repressor function. I'm going to show you proteins that bind to DNA. We've also used CRISPR systems based on deactivated Cas9. You can use small RNAs, invertases, any biochemistry that you want where when you turn it on, it then turns something off. And the only important thing is that both the inputs and the output have to be transcriptional in that the inputs have to be promoters and the output has to be a promoter. And that's what allows you to combine the different circuits in order to build up more sophisticated programs. So one of the key problems is that in order to build a large program, unlike electronics where you can define one circuit and then just reuse that circuit over and over again, in genetic circuitry, you need to remake many versions of the circuit so that they don't interfere with each other because it's all based on biochemistry that's pushed together in the cell. So we've been doing this for a lot of different systems where at the core of the system we have a repressor. So in this case, we were using a repressor that was based on the TEDR prokaryotic repressor. We then used bioinformatics to identify a large library of alternatives and had to screen them very carefully in order to identify a subset that don't interact. And so this is showing a series of different repressors and the promoters they interact with. And you can see that after this screen, this gives you a set of repressors, each one of which can serve as the core of a NOR gate, where then all of those NOR gates don't interfere with each other. And so once we built these different repressors, we put them into the context of gates, and then started trying to be able to build more sophisticated circuits out of these core gates. And so we would have a set of different repressors that we had proven they all don't interact with each other. We then have sensors, which are leading to the interaction, leading to some sort of environmental stimulus. And then if you want to build up a circuit, so this is a 4-gate circuit that acts like an exclusive OR gate, then the trick becomes identifying those repressors that connect well to each other, and then you have to divide the repressors up in order to make a linear piece of DNA that encodes that wiring diagram. And so now this is a linear piece of DNA that has 3 repressors, the 2 sensors, and then an output, and it should, in the way that it's connected, create this exclusive OR function. And so once we built all of these different gates based on the repressors that interact with each other, we just automatically started to assemble them into different genetic circuits and looked at how well we were able to create whatever circuit we wanted without any additional information. And so the result of that experiment is shown here, where in this case we tried to make 16 different circuits based on that library of gates. And there was different levels of sophistication in each circuit. And what I'm showing in the gray and the black is what the function should be, and then in the red and the blue what the experimentally measured function was. So in this particular case, this circuit behaved as we would expect. But then if you look here, the circuit completely failed. And if you focus on those particularly bad failures, you'll notice that a lot of them are the most sophisticated circuits that we were trying to build, where we were unable, just by snapping together the gates that we had constructed, to be able to produce the functions that we wanted. And so this year we published in Nature Methods a review paper that captures all of the different ways that over time we figured out how these circuits are failing and why we weren't able to put together the gates and have them function properly. So using all the different failure modes, all the ways in which the combination of gates can possibly go wrong, we went in and found a couple of failure modes that were particularly predominant. The first is that we had a lot of problems with transcriptional read-through. And so if you look at the way that we divide up the gates in order to create a circuit, there are these terminators that have to shut off transcription after a gene. And one of the characteristics really in synthetic biology is that you have this very punctate transcriptional program that you're trying to control. You have an extremely strong promoter, a small gene, and then you need to stop transcription very dramatically or else it reads through into the next gene. And we were seeing that our terminators weren't very good, and so that was leading to a lot of these failure modes. The second problem we found is that when we divide up the gates from how we characterized it into the final circuit, often we'd have to split the gate. So in this case, we have a promoter and a gene that are part of a gate. The gene goes here, the promoter goes way down here. And so this promoter in this new context will often function differently. And we found that if you add ribozymes then between the promoter and the ribosome binding site, that was one way that you could get the promoter to be less context dependent. So we figured out these different things. We had to go through these really painful mining exercises where we would do things like screen 500 terminators just to find a subset that were really strong so that we could shut down transcription without having homologous recombination by reusing the part. Ribozymes, we found one and published one that was good, but then we need 20 or 30 alternatives that are equally functionally good so that we can put one behind each gate. And so after we did that, we could create a new library for each one of our repressors. We then have a very strong and unique terminator to shut down transcription. And then upstream, we have a unique and very strong ribozyme so that any promoter can be put in front of the gate and it functions approximately the same. And so we refer to these as the insulated gate, where they're the next generation of these fundamental gates, where we added a lot of parts to the system in order to make it where each gate really functions in exactly the same way, no matter what circuit it's participating in. So after we did that, we reassembled the gates into the circuits. And what we found is now every one of these circuits behaves exactly like we had predicted. And so all of the ones where we were seeing difficulty before, we now are able to get the expected function out. And this is with no downstream fiddling. So it's just taking those gates themselves and connecting them in such a way that they reproduce the wiring diagram that you're trying to have it function as, and not mutating it after the fact in order to try to get that function back. And this was just from taking these gates that were failing and just being very careful about isolating those gates from all of the context effects that happen when you shift their position around. So as soon as we had this, we decided to get a bit more ambitious in what we were doing. And so because we had made these gates that seemed to be simple enough where you could snap them together in different configurations and be able to predict their function, we thought that that might be simple enough to have a computer do that process for us. So we created a software package in collaboration with Doug Densmore at Boston University. And this is going to be released publicly in the next couple of months. We call it Cello for cellular logic. And it utilizes Verilog, which is a circuit descriptor language out of electrical engineering that can capture in a hardware-independent way any particular circuit function that you want to create. And so you write the circuit function that you want in Cello. It then goes behind the scenes and takes those insulated gates that we've constructed, figures out using tools from electrical engineering what the wiring diagram is that's best for the circuit function you want. Then it goes to the gate database and maps the different circuits onto the wiring diagram. It then creates a linear piece of DNA, which then is the output. And you can either build it yourself or send it to a synthesis company to construct. And so this is a entirely black box. You give it the circuit you want. It gives you the DNA sequence back, utilizing only those gates that we've constructed to be compatible with this circuit design language. So there are a lot of algorithms behind the scenes that are going into figuring out exactly how to put these different gates together. Just to give you an example of one of those algorithms, one of the challenges is that each one of the different gates has a slightly different input-output function. And so you have to figure out what gate is the best at each location in the wiring diagram. And so one example of an algorithm that we use is just searching the space by putting the different, this is not going to be coordinated, by putting the different gates together and then figuring out where you have some score for how good the circuit is functioning, and then you have it converge onto some final construct which then assigns a gate to each location in the wiring diagram that gives the best possible function out of the back. That then leads to a particular DNA sequence that comes out. And so we've now tested this, and these are entirely compiled, still very simple programs, but now getting up to 6 gates, where now everything is automatically compiled. So you write the program that you want, you push enter, out comes a DNA sequence, you test that DNA sequence for function, and we've been finding that we can, more often than not, actually get the circuit function that we are trying to achieve without this after the effect fiddling. And it really came down to having these insulated gates. And now that we have these, we're really starting to build much larger programs, much larger circuits than we've done before that have 20 or more different transcription factors that are part of a much more sophisticated design, where we're either using this program to build the entire thing at once or breaking it into submodules and designing and testing each one of those individually. Okay, so that's where we are with respect to building synthetic circuitry. We now have the software in place where we can take a set of sensors that we characterize independently. The levels that those sensors achieve get fed into this design software. You put in how you want the cell to react to the different combinations of inputs. It then figures out the wiring diagram and assigns the transcription factors. So the next thing that you have to do is have somehow where the output of those different circuits controls the various actuators, or the cellular responses that you want the cell to have when the different combinations of inputs are in the correct state. And so I'm going to talk now about what we're doing with respect to gaining control over these actuators in the cell. So one of the challenges is that when you want to try to gain control over something that the cell is doing, if you're trying to do it over some natural function that the cell already controls, the challenge is that the cell itself has all of these regulatory mechanisms to make sure that that function is only turned on under those conditions that are relevant for that function for the cell and not necessarily relevant for what you're trying to do when you're getting synthetic control over it. And so we started to take a general approach where we wanted to be able to have it in the future where if we're trying to do genomic engineering and build up a complicated function, we wanted to be able to take things that the cell could do and just snap it into our design. So for example, if we want the cell to secrete protein, we wanted to be able to just take all of the genes associated with this, say, secretion needle, and take a unit of DNA and snap that in, and then now the cell can secrete protein. Or if you want to be able to fix nitrogen, for example, we wanted to be able to take a unit of DNA that has all of the necessary genes to fix nitrogen and snap that into the program, or a natural product, or anything that the cell can do, really have this kind of modular mix and match ability to take these different functions and combine them as part of building up a larger program. And so we started to get interested in gene clusters as a mechanism towards that, where gene clusters are convenient because they take all of these different cellular functions, and all of the genes that are associated with that function occur in a cluster together in the bacterial genome. So for example, in the case of protein secretion, in Salmonella, there's a 35,000 base pair region of the genome that has all of the genes that build that structure for protein secretion. Same with nitrogen fixation. There's also many natural products, for example, that have gene clusters that have all the genes necessary to make that natural product. And so what we were imagining is that we could take all of these different gene clusters and mix and match them as part of building up those functions. But there's a challenge in doing that, which is that each one of these clusters is under very sophisticated regulatory control by the cell, so that function is only turned on under the conditions where that function is relevant. And this can be very sophisticated. So when you look up a particular gene cluster in, say, Nature Reviews Microbiology, and you look at how the cluster is organized and regulated, you have this very simplistic view, this kind of minimalist view of how the regulation is affecting that cluster. But we know that in natural genetics, there are layers on layers of control that are complicating that picture. So for example, you might have a cluster of genes associated with the function that has a nice internal regulation shown in red, but that internal regulation is highly embedded in the global regulatory network of the cell. The way the genes and the parts are encoded is extremely non-modular. So even simple things like translational coupling, you'll have a ribosome binding site upstream of a gene that's in the middle of another gene. And so this complicates your ability to be able to engineer the system. There are lots of regulatory parts that have multiple embedded functions. So things like promoters often occur within open reading frames. Promoters are often pointing the wrong direction for no apparent reason. And as you learn more and more about even the most well-characterized gene clusters, it's just like peeling an onion, where you're constantly learning new things about how it's being regulated. And then for many of the most interesting functions, they're not really well characterized. And so what we had to do is somehow think of a method where we could go in and wipe out all of the native regulation for a cluster so that we could implement synthetic control from one of these gene circuits. And so in order to do this, we go through a process called refactoring, which just is a systematic way of stripping out all of the regulation for a natural cluster. So we take a gene cluster that we want to get control of, and we then remove all of the non-coding DNA and all of the regulation that might be encoded within that. We take all of the open reading frames and we codon randomize the open reading frames, which means that we select codons to try to create DNA sequences that are as far away as possible from the wild type sequence. Idea being that we want to create genes that don't have any internal regulation, so they're inert to everything except encoding the amino acid sequence we want. We then reorganize those refactored individual genes that have been codon randomized. We organize them into artificial operons. We pick synthetic regulation. And then we use phage promoters to control transcription so that we can then build a controller that has the synthetic sensors and circuitry. The output is a phage RNA polymerase, and that output then turns on all of the necessary operons associated with function. And so we've gone through this process now for a variety of different gene clusters in order to try to be able to replace the control. And so this is an example of one of the gene clusters we've done a lot with. This is a gene cluster that encodes nitrogen fixation, which is important in agriculture. And I could give a whole talk on that, but from this perspective, we just want to be able to transfer it between organisms. We went through this process of refactoring, and remarkably, when you're done with this process, it works, where we take all of the different genes, all the native regulation has been stripped out, the system has been modularized into a set of parts, each of which has an assigned function, and then importantly, the system is turned on by phage promoters so that you can then use a controller that has some sensor, in this case just an inducible system, that then induces phage polymerase, which then turns all of the necessary genes on so that you then fix nitrogen when the controller is on. And so what that allows you to do is then very simply swap the regulation for different synthetic controllers encoding different sensors and circuitry. So this is an example where we just have an IPTG inducible system driving RNA polymerase, and so we get nitrogenase activity. If you want to put it under different control, you just swap in a different controller that responds to a different sensor, and you get the same activity out. And then if you want more complex control, you can then put circuitry onto the controller, and now you only get nitrogenase activity in the right conditions that the circuitry is dictating in how it's responding to the various sensors. And so this is where we've then taken this gene cluster that encodes nitrogen fixation, stripped out all the native regulations so that you can put it under synthetic control. And we've been going in and doing this for a variety of different functions, and it works each time. It's kind of a torturous process, but you gain this control. So this is an example of type 3 secretion. So this is a 35,000 base pair gene cluster that builds this, this big needle-like syringe that goes through the inner and the outer membranes and pumps protein out of the cell. And so again, we went through all of the same process where we could refactor this to create a minimal gene system with a controller that then integrates various inputs and tells the system when to turn on. And when we go through that process, then now when we want to turn on protein secretion under conditions that repress the wild-type gene cluster, we can then turn it on when we add the right inducible conditions. And so this allows us then, with this refactored gene cluster for protein secretion, this becomes a unit of DNA where now you can put that unit of DNA into the cell. It has all of the genes necessary to induce this protein secretion device, and that becomes then another output that the cell could do. And we've started to build up more complex systems where we combine these different functions. So this is an example of dual control, where now we have circuitry with different RNA polymerases that don't interact with each other. And then under conditions where you turn on one of the pathways, it turns cells red. Under the conditions that turn on that are different, it then causes the cells to turn green. And so the logic that's in this controller and the way that it's integrating the sensors switches between these two pathways. And you can see that here. And so we've been snapping together these different types of functions to build up these more complex programs that we're putting in the cell. Okay, so I started out by describing the different ways that we create controllers. So we have the synthetic sensors and circuitry that tell the cell when to respond by turning on different things that the cell can do. When we want to take a system that the cell already has, like protein secretion or a natural product or whatever it is, we have to go through this process of stripping out the native regulation and putting it under control of synthetic regulation so that we can induce a RNA polymerase that then turns on that particular function. And so as we did that, we started to notice that it would be valuable as an intermediate step to be able to control the activity of the various RNA polymerases that were needed to turn on these different systems. So you can imagine in this hypothetical example, we have 4 different wires that go from the circuitry to different cellular responses. This would require 4 different RNA polymerases that are orthogonal to each other and don't interact. And so you have this problem where that becomes kind of difficult for the cell to manage in practice. It's also difficult to deal with as you're building because those genes are very large. And that's captured here. So this is, you can imagine if we have a controller and it has these different RNA polymerases, if we're expressing 4 complete RNA polymerases, each of which is 3,000 base pairs, now we have 12 kb, that's just the 4 different polymerases, to try to turn on 4 different pathways. The size of the DNA is a problem, but more importantly, if you're under conditions where you express all 4 of those polymerases simultaneously, that can be very toxic to the cell. And so in this case, if you express the 4 polymerases, then if it crosses this threshold, then the cells die because it crosses that threshold. And so what we want to do is be able to set some total amount of transcriptional activity that those 4 polymerases share that are below that threshold. Am I running out of time? Okay, I will wrap it up. So the way that we deal with this is that we have we took the phage polymerase, we cut it into multiple pieces, and so that we could just express 4 different fragments that have different promoter activities so that we can set the core fragment activity constant, and then it shares that resource between the 4 different polymerases. We also have additional regulatory things that we can put on where we have an upstream piece that has to be expressed for activity, and then we have a fragment that doesn't bind to a promoter that can act as a negative regulator. And so when we do this, we can then set the total amount of polymerase activity that then 2 different outputs from the circuits have to share in order to be able to turn on the 2 different activities. And if we set it high, then those two different outputs share that resource, but they never get above the toxicity level. And then if we use a low amount of core, they then share the resource and go below. So with that, I'll conclude. So I took you through the different ways that we're trying to build up more complex designs, and where we're trying to create the resources in place where we can build circuitry that is more reliable and more sophisticated that we can do, currently with the idea of pushing that up to the scale of the cell as a whole, and where we're able to put together larger genetic programs by taking these different functions that the cell can do, stripping out the native regulations so that we can put them under synthetic control. And then finally, where as part of building up the big programs, being very careful about controlling the resources so that it doesn't get beyond a point where the cells die. So with that, I will conclude by thanking my lab. So we have a number of different collaborators that are involved, and I'm happy to take any questions. Thank you.
Great. Thank you very much. So we can have a couple of questions.
Chris, I'm very interested in your gene cluster for nitrogen fixation. What's the efficiency compared with the wild type? It's almost same, or it's still some distant?
So if you look at just within Klebsiella, it's about 60% of the wild, so about half, roughly, of the wild type activity. That just came out last week.
Okay, so any way that you are working on to improve the efficiency?
So we're not actually working on it anymore with respect to within Klebsiella, but we're very much focused now on moving that to organisms that don't have that activity, that associate with different crops. Mm-hmm. So the advantage of the refactored version, specifically of the nitrogenase, is not getting more activity. That's not kind of what's useful there. But rather, when you have to move to a new organism, and you have to do things like change all the ribosome binding sites and all of the promoters at once, that is a move in terms of in genetic space that you can only do with the modularized system. And so if we want to move it to some obscure organism that uses a different ribosome binding site, has a different 16S RNA, you can swap all of them at once. And so once we got good enough with activity, that's where we're focused now. Yeah. Yes.
Anyone else?
Yeah, wonderful talk. One question I have is, what kind of host cell system do you use to test your hypothesis? How do you eliminate those variables when you do biological experiments? Because when you transform the DNA, the DNA quantity, those kind of things, how do you make sure the data you obtain reflect your design?
I'm not sure I understand the question. Do you mind?
So when you do biological experiments, when you transform the DNA, different quantity of DNA might give you different results instead of the design of your circuits give you the results. How do you remove those variables of your experiments?
Right. So for the most part, we're working with bacteria and we're working with plasmid systems. And so no matter how much DNA you transform, they more or less go to a certain concentration of DNA in the cell. There's some variability there. That becomes a really big problem for mammalian cells, where if you're transforming a plasmid in, you can have very dramatic changes in the amount of DNA. And that becomes a big problem. So when we've done work in mammalian cells, that's been a significant effect. In terms of the bacteria, it's less of a problem. And you could always go to putting something in the genome, which very stringently controls the amount of DNA.
But even for bacteria, different growth phase of the bacteria might have different results. How do you control that?
For the systems that I'm showing here, that's not so much of an issue, but sometimes it does come up. And one of the things that we're doing now is trying to identify situations where that becomes a problem and then go back and try to come up with an engineering response. So for example, one of the things my lab's very focused on at the moment is creating systems that are copy number independent. So you can imagine taking a promoter and putting all the necessary regulation behind that promoter so that when you drop it in the genome or at a low copy plasmid or even a high copy plasmid, it still produces the same amount of expression. And so long as you can define the type of thing you want to be robust against, I think we're getting sophisticated enough where you can come up with an engineering solution for how to create a system that's robust in that case. And that's similar to what we did for the circuits, where once you define why they fail, you don't have to just accept that that's biology. You can then start to think through, well, how do we insulate the system against that failure mode and go from that point.
Thank you.
For the interest of time, we'll stop the questioning session here, and let's thank Professor Chris Voight again for his wonderful lecture.